The overall goal of the following experiment is to assess the level of colocalisation of internalized bacteria Neisseria meningitidis with the cytoskeletal protein α-actinin. This is achieved by infecting cultured human brain microvascular cells with an overnight culture of bacteria for 3 to 8 hours to allow the bacteria to enter the target cells. As a second step, cells with intracellular bacteria are washed, fixed, and then permeabilized, which prepare the infected culture for immunostaining of internalized bacteria and intracellular α-actinin. Following the Confocal Imaging and application of colocalisation software, images of the intracellular colocalisation can be observed and quantified. Results are obtained that show meningococci expressing OPC and outer membrane protein can specifically interact with intracellular α-actinin based on confocal microscopic visualization and by quantitative colocalisation analysis.
Hi, I'm Isabel Murillo from Professor Mumtaz Virji's lab at the Department of Cellular and Molecular Medicine at the University of Bristol. Today we will show you a procedure for estimation of the level of colocalisation of intracellular bacteria with cytoskeletal proteins. We use this procedure in our laboratory to study the interaction of Neisseria meningitidis expressing an outer membrane protein, OPC, with the cytoskeletal protein α-actinin. So, let's get started.
This work needs to be done in an adequate safety laboratory. 2 days prior to the immunostaining procedure, inoculate the bacterial stain of interest on brain-heart infusion agar plates supplemented with 10% heated horse blood. Grow overnight at 37 degrees Celsius in a 5% carbon.